This is 0620 Biology, Paper 4, Variant 1, October-November 2019 session. Alright, starting with question number 1. All living organisms are placed into groups according to their features. Meropods are one of the main groups of arthropods. A. State 2 features of meropods that can be used to distinguish them from other arthropods. Alright, so there are a few features, distinguishing features, and some of them can be the fact that they have numerous body segments, and similarly they have many jointed pairs of legs. Okay. Figure 1.1 shows that there are 4 main groups of arthropods, group 1 being meropods, and there are 3 other groups. B. State names of 2 of the other groups of arthropods. Right, so the other 3 are insects, arachnids, and crustaceans. We only need to write 2. So I'm writing C. Meropods can be classified into 4 classes: 1, 2, 3, and 4. Figure 1.2 is a dichotomous key that can be used to distinguish the 4 classes of meropods. Right, so this is a key describing meropods, and here there are only 2 groups, with eyes and without eyes, and we're supposed to use the key to place them into classes 1, 2, 3, and 4. Complete table 1.1 by using the key in figure 1.2 to classify the 6 meropods in figure 1.3 into the 4 classes. Alright, let's start with the first picture and see what we've got in the key. So we're supposed to see if the organism has eyes or not. This one doesn't seem to have any eyes that are visible, so we're gonna go with group with no eyes. And does it have hair on most body segments or not? Let's see. This seems to have these tiny hair, so which means this species J is going to be classified in class 1 because it has no eyes but it has hair on its body segments. So 1 in class 1 we're going to place J. Right, let's move on to the species K. This one, alright, it has eyes present as we can see over here. These are 2 little eyes, and we need to see if it has 1 leg on each side of each body segment, or does it have 2. Alright, so as we can see over here, this segment has 2 legs on one side, 2 on both sides, which means we're going to have to go with class 4. So we're going to be placing K in class 4. Similarly, species L, alright, so it doesn't have any eyes, so it's going to be in either of the first 2 classes. And let's see if it has any hair or not. It doesn't seem to have any hair, so we're gonna be classifying it in class 2. Alright, and M, alright, this has eyes, so it is going to be in either of these classes. And let's see how many legs it has on each side of a segment. This has only 1 leg, so it's going to be in class 3. Moving on to species N, right, it has got eyes, and let's see how many legs it has. Right, as we can see over here, it's quite small, so it might be difficult, but we can make it out that there is more than 1 leg. There are actually 2 legs on each segment of its body. So species N is going to be in class 4 with eyes and 2 legs. And the last one is O. It does have eyes, which means it's going to be in either 3 or 4. And it also has two legs on each side of each segment. So it is also going to be in group 4. Alright. Part D, figure 1.4 is a photomicrograph of the meropod Elphoria virginiensis. 1. State the genus name and the kingdom name for the meropod in figure 1.1. Alright, so in a scientific name, the first name is always the genus and the second being the species. So its genus name is Floria. Right, and as we can see over here, this is an animal, quite obviously. So it belongs to the animal kingdom. Alright, A. virginiensis releases the poison cyanide when it is attacked by predators. Cyanide stops enzymes in the mitochondria from functioning, suggests why cells die if the mitochondria do not function. Right, so mitochondria are powerhouse of the cell, which means aerobic respiration takes place here. So if the mitochondria are not working, that would mean that aerobic respiration is no longer taking place, and hence the cells do not get any energy to perform, so they die. Okay, question number 2. Phloem is used to transport sucrose and amino acids in plants. Sucrose is a carbohydrate. A. Describe the uses of carbohydrates and amino acids in plants. Alright, so carbohydrates have many uses, and some of them can be in respiration, for energy, to produce glucose, as we know is one of the products in respiration, and it can also be stored as starch. Alright, so we've mentioned two uses of carbohydrates, and it would be wise to mention two other uses of amino acids since the question is of 4 marks. So we're going to be distributing the marks equally between the two groups. So amino acids, we need amino acids to make proteins, and those proteins can be used in many functions of the plant, like being used as enzymes or for growth. So making proteins. Alright, B. Figure 2.1 shows a diagram of a plant. The arrows point to the circles containing magnified cross sections of those parts of the plant. Alright, so these are the three pictures. The first picture is a magnified picture of the upper surface of the leaf. The second one is another picture of the stem, and the third one is one of the roots. Label the positions of the phloem in each of the three magnified sections in figure 2.1. Use the label line and the letter P for each section. Alright, so we're supposed to be labeling these for the phloem vessels. Alright, so another rule for labeling is that it would always be better if you do not use arrowheads. Simply draw a line. Alright, so as we can see over here, the outer part of this bundle is phloem. So I'm gonna write P over here. We don't need any arrowheads. Just simply draw a straight line with using your ruler, and that's it. And similarly over here as well, we have the outer part as phloem in the vascular bundle, and here also this outer lining is the phloem vessel. C. Arachnids are used by investigators to discover how plants transport sucrose. Figure 2.2 shows an aphid with its mouth parts inserted into a plant stem to feed on the liquid in the phloem. Okay. A plant was put in a dark cupboard for several days. 4 aphids A, B, C, and D were then placed on the plant in the dark cupboard as shown in figure 2.3. Okay, so this is a dark cupboard. And as you can see over here, we have 3 of these aphids on the surface or the top parts of the plant, and D is relatively lower on the stem. So this dark cupboard means, alright, so we are putting it in dark cupboard for several days, so it would not be exposed to any sunlight or any sort of light for that matter. So it's not going to be able to carry out photosynthesis. Okay. Part 1. Immediately after the aphids were placed on the plant, it was observed that all of the aphids ingested the same volume of liquid from the phloem. Aphid D ingested the highest concentration of sucrose. Explain why aphid D ingested the highest concentration of sucrose. Alright, so as I mentioned earlier, the plant would not be able to carry out photosynthesis, which means no production of glucose. And now, since the plant needs the glucose or carbohydrates, the plant is going to be using the already stored sources in the roots in the form of tubers. So what's going to be happening is the stored starch in the roots is going to be converted to sucrose to be transported from these roots to the other parts of the plants and to all of the leaves as well. So what happens is when that stored starch gets converted to sucrose and moves up the stem, the first what is the first organism that is able to access this sucrose, and that is why it is able to achieve the highest concentration. And then the remaining sucrose gets distributed, which means the amount or rather the concentration of that sucrose is going to be distributed and less than it was over here for aphid D. So ABC hence get a much lower concentration of sucrose. We need to mention all of the points which we've discussed above. Alright, it is quite crucial that you do mention the process of translocation, since that is the main concept being questioned over here. Okay, part 2. Many crop farmers try to prevent insects such as aphids from damaging their plants. Describe how modern technology is used to reduce damage to crop plants by insects. Okay, so we can use genetic engineering from biotechnology to insert some genes in those plants which can make them resistant to pesticides. But aside from that, we can use a few genes that let's say an example of Bt toxin, which when inserted into cotton plants, it causes death of any insect that tries to eat it, and so the insect would naturally not be attracted towards those plants, and the farmers would then not need to spray any pesticides. So this is how the use of pesticides and also insects can be reduced by using biotechnology. Okay, so okay. Part 3. Other insects are useful to crop farmers. Give one example of how insects are useful to farmers. And the only situation that I can think of is by pollination, so that the insects can transfer pollen grains from one plant to another for reproduction. Okay, question number 3. Kidneys are important for excretion. A. Explain the purpose of excretion. Well, that is removal from organisms. You can write the whole definition of excretion in such questions where you're supposed to explain um, a process. This could have been osmosis or diffusion or active transport. Okay, part B. Sketch a simple diagram of a human kidney and its ureter. Label the cortex, medulla, and ureter on the diagram. Okay, so we're going to be drawing a kidney over here. Okay. So this is the ureter, and over here we have the outer layering as cortex and the inner part of the kidney as medulla. C. Figure 3.1 is a photomicrograph of a glomerulus in a kidney. 1. Describe the structure of a glomerulus. You may refer to figure 3.1 in your answer. Okay, so glomerulus is the tangled up structure in the kidney where filtration of blood occurs. So we can write it as okay. These are the capillaries here need to be quite thin for so that molecules like water, salt, and glucose can escape out or move out easily by diffusion. So they are only one cell thick, and those are the features which you can mention since we're only supposed to be describing the structure of the glomerulus. 2. Describe the role of the glomerulus. Okay, so filtration occurs here. And what happens during filtration is that molecules like water, glucose, salt, and urea move out. Okay. D. Table 3.1 shows some comparisons between a human and a mouse. Alright, so we have a table over here. So this is a comparison between a human and a mouse, mentioning body mass, kidney mass, water intake, water reabsorption, salt filtration, and reabsorption of both organisms. 1. Table 3.1 shows the salts are reabsorbed in kidneys of both humans and mice. Describe how salts are reabsorbed against a concentration gradient. So the only process which can absorb ions or particles against a concentration gradient is active transport, and since we are supposed to be only describing that process, we're going to have to write the whole definition of active transport. But first we need to mention by active transport. Alright, so here I've written the whole definition of active transport. Scientists stated a hypothesis: humans and mice absorb salt at almost the same rate in relation to the size of their kidneys. Determine whether the data in table 3.1 supports the hypothesis and show your working. Okay, so the hypothesis states that they absorb salt at the same rate in relation to the size of the kidneys. Alright, so this means we're supposed to be calculating the rate since we are supposed to see whether this hypothesis is right or not. So the rate, we're going to have to be dividing the amount of salt reabsorbed with the size, since the rate is relation to the size. So reabsorbed salt divided by size of the kidney. Okay, so for humans the calculation is going to be 575 grams divided by the kidney mass, which is 320. So we have 575 divided by 320, and this gives us the answer of 1.797. And for mouse we have 0.551 grams of salt reabsorption divided by the kidney mass, which is 0.310. Okay, So this gives us the answer of 1.778. Okay, as we can see over here, the values are almost the same, which means the hypothesis is correct. Okay, yes. 3. Table 3.1 is 3.1 also shows that water is reabsorbed into the kidneys of both humans and mice. State the name of the process that mammals use to reabsorb water. Alright, so the movement of water occurs by osmosis. 4. Water and salt are reabsorbed in the kidneys. State the name of one other molecule that is also reabsorbed by kidney tubules, and that is glucose. Glucose can be one of the molecules which is reabsorbed. Okay, question number 4. The A. The body has defense mechanisms to protect it from infection. Outline the body's defense mechanisms. Alright, so I'll start with the chemical and mechanical barriers. And the hydrochloric acid in stomach, which kills any bacteria as chemical barrier. And then we have another system. You can just start with white blood cells. So there are phagocytes and lymphocytes. I'm going with lymphocytes. Alright, so lymphocytes produce antibodies. So once lymphocytes have created antibodies for a particular pathogen, some of those antibodies would then be saved in the body as memory cells, and then this provides active immunity since if the infected person gets those pathogens inside his body for the second time or again, he will be ready to defend his body against that particular pathogen since these memory cells would be ready to end it. B. State the name of a type of drug that can be used to treat bacterial infections, and that is. Okay, moving on to question number 5. Figure 5.1 shows an angiogram of a heart before and after treatment of coronary heart disease, CHD. An angiogram is an image of the blood flow through the blood vessels of the heart. Okay, so before the treatment, we can see over here the blood flow is is quite different from the blood flow of the arteries above this area. However, after the treatment, it is uniform. So A, the arrow in figure 5.1 shows the position of a blockage in blood vessel. State the name of the blocked blood vessel, and since this this is a coronary heart disease, the blockage is going to be in the coronary artery. Okay. Part 2, the blockage is caused by a blood clot. Describe how a blood clot forms. Okay. So platelets in the blood release there is a few chemicals which then convert fibrinogen, which is a soluble protein in the blood plasma, and then that protein is converted to fibrin, which is insoluble, and that fibrin then forms sort of like a mesh with red blood cells and forms an insoluble tangle which prevents the blood from getting out of the body, and it also acts as a barrier against pathogens from entering into the body. Okay. Part 3, state the name of a drug that can be used to treat coronary heart disease, and one of those drugs can be statin. You can write any over here. Many health specialists think that the risk of coronary heart disease can be reduced by doing regular exercise. A long-term study of a large group of women was used to test this hypothesis. The women were between 35 and 45 years old at the start of the study. Every 2 years, the same group of women were asked how much they were exercising. After 28 years of researchers analyzed their data, they calculated that the average time spent exercising per week by each woman. They put the women into categories determined by how much exercise they had done. For each category, they calculated the number of women who died from coronary heart disease. Okay, and here is the data provided in the form of a graph, bar graph. 1. Calculate the percentage decrease in the number of CHD deaths per 1000 women. 10,000 women between those who did not exercise and those who exercised for 4.1 to 10 hours a week, using the data in figure 5.2. Alright, so the number of CHD deaths per 10,000 women who did no exercise. Okay, so this is the group with no exercise, and we can figure out the value over here. As we can see over here, we can figure out the value used to represent 1 unit. We can divide 0.2 with 5, since here there are 5 blocks having the value of 0.2. So this gives us 0.04, which represents each unit over here. So we're going to be adding 0.4 times 2, since there are 2 boxes over here, with 1.8, which gives us the answer of 1.88 deaths per 10,000 women who did not exercise. So this value is 1.88. And the number of CHD deaths per 10,000 women who did exercise from 4.1 to 10 hours per week, and this is the group with the least value, and this is only 1 unit, which we earlier calculated was 0.04. Okay, now so we're supposed to be calculating the percentage decrease, and we can calculate that by working out the difference between the two values, which is 1.88 minus 0.04, and then we're supposed to be dividing. Okay, so we divide this with 1.88, so we get the answer as 0.9789, and then we're supposed to be multiplying this with 100. Okay, so we get the answer over here as 97.8, which if we round it is equal to 98. So here we have the percentage decrease. Now, health professionals wanted to use the results of this study to encourage the whole population to take more exercise, discuss the arguments for and against health professionals using the study in this way. Okay, so we're going to be using the information about this study, how this study was designed, and the results in figure 5.2 in our arguments. Okay. So we need to collect data which would give us reasons for and against this investigation. So one of them can be that increasing exercise decreased CHD level in women. And as we can see over here, women only exercised a few hours, and still it did cause a significant amount of decrease in their disease. So I'll be mentioning those points along with some data which can cover me 1 mark. Okay, so increasing exercise. So I just wanted to add a point over here that whenever we have qualitative figures like this one graph and we are supposed to be describing or discussing, quoting the data would always score you some marks. So just go with that. It's quite easy and you don't need to know any theory about that either. So we can start with how there were 1.88 deaths per woman for 10,000 women for those who did not exercise, and only 0.04 cases for those women who did exercise. So okay, so these are the 3 points that I have mentioned for the study, and now I'll have to mention 2 points, two remaining points, so I can score all 5 marks. So those 2 points are going to be against the study. As we can see over here, this study was only conducted for women, so that can be 1 point. This is not going to be fair since we do not have any men or young children or any older women either. So that is going to be 1 point. Well, actually, those are 2 points. One being that there is only one gender, and the other one can be that the age group is limited. So only okay. This part is completed. C. Exercise can cause heart rate to increase. Explain why exercise causes an increase in heart rate. Okay, so for exercising, muscle contraction increases, and for greater muscle contraction, we need more oxygen to create energy by respiration. And for transporting that increased amount of oxygen into our cells, we need a faster blood supply. So, okay, question number 6. Fish are an important part of a balanced diet for many people. A. Fish are a good source of vitamin D. State one effect of a diet that is deficient in vitamin D. Well, it can cause weak bones. Part B. Many fish stocks have declined. Alright, marine ecologists considered 3 topics shown in figure 6.1 when they discussed the decline of fish stocks. Okay, so the reason why fish species became endangered, risks to a fish species when population decreases, and methods for maintaining stocks. Okay, describe the key points for each of the 3 topics in figure 6.1. Okay, so we're going to be giving reasons. Let's see how many. Okay, so this is a marks for question for 6 marks. And by distributing those marks equally, this would give us 2 points for each of the objectives over here. So let's start with reasons why fish species have become endangered. Well, this can be a result of overfishing or humans hunting them. And another point which I'm going to be mentioning over here, well, overfishing, it causes food chain disruption of food chain, and this can then result in decline in multiple fish stock. And the second point I'm mentioning over here, it can be diseases in the fish stock. Alright, so here are 2 points for the reason. Alright, so the risks to a fish species when population decreases. So this can lead to less variation and also extinction of those species. Alright, I've mentioned 2 points over here, and the last one says methods of maintaining fish stock. That can they can be maintained by educating people on the importance of fish stock. So and the second point can be providing quotas or implementing quotas or restoring fish. So this is how we can control this situation, and I've mentioned 2 points for each of them, which covers the 6 marks. Moving on to question number 7, figure 7.1 shows part of the nitrogen cycle. Okay, A1 state 2 ways that nitrogen fixation can occur. Okay, that can occur by lightning. Alright, state the names of the processes A and B. Okay, let's see what process A is. Alright, so conversion of nitrate ions in soil to nitrogen in atmosphere, that is denitrification. And B is, what is process B? Okay, so process B is conversion of compound X to nitrate salts. Let's see what compound X is. Right, so amino acids are converted to proteins in animals and plants by protein synthesis in their cells, and then those proteins are then deaminated to plant and waste. Okay, deamination occurs and waste is produced, and then that waste is converted to compound X. Alright, so this waste is going to be decomposed by bacteria or decomposers in the soil, which are then going to be converted nitrogen or proteins in the waste to ammonium ions, which should be compound X, and then those ammonium ions can then be converted to nitrate ions in the soil by process B, which is nitrification. Alright. 3. State the name of compound X. Over here, this is the ammonium ions. Define the term deamination. Alright, so we're supposed to be writing the definition over here. To form urea. Alright. Moving on to part B. State the name of the structure in cell where protein synthesis occurs. Well, that structure is ribosome. C. State the name of an enzyme that digests protein. That can be protease. You can be specific over here and mention any other enzyme, but I'm going with protease, and we're done with this paper.